Welcome to the first meeting of 2023. It's nice to see 19 people in the room and 23 people online. Really nice. So maybe those figures will go up, but really nice for you all to join us tonight anyway. Thank you. Soon we'll be hearing from Dr. Julia Schroeder about the sparrows' intimate lives on Lundy. But for the moment, I want to see if anybody would like any announcements to make any, and I know that Neil wants to make an announcement, so I will just pass the mic over to him. If you want to come up here, Neil.
I will indeed, yes.
Right.
Good evening, everybody. Apparently for a few years there's been an attempt to run a national red kite survey in January every year, and this has then just been drawn to my attention, and nobody's doing anything in Berkshire, or indeed I think either Oxfordshire or Buckinghamshire so far. It is, the methodology is to count birds going into winter roosts. And so I thought, having had this drawn to my attention, I'd have a quick look at large birds, see where our winter roosts are, and there's none on any of the recent records. Now, it may be because as we're a long thin county, that all our birds are actually roosting over county borders. Certainly the nearest one to me is, as I'm aware of, or I haven't gone to see it for years, actually in Oxfordshire. But if anybody knows of any red kite roosts around the county, can they drop me an email on the conservation@parksoft.org email address? And also, if you are willing to spend an evening going and counting kites going into a roost, if we can find a roost, if you could also please drop me a line, or if you're here in person, see me at the break. Thank you.
Thank you, Neil. Right, so I guess that's probably all in terms of announcements. I would just like to mention the next two meetings. Rob Robinson on the 18th, 2 weeks today, is talking about the conservation value of ringing. And on the 1st of February, Dr. Mark Eaton will be here to talk about nurturing, hang on, sorry, just a moment, put my glasses on. Sorry. Monitoring rare breeding birds. Okay, so let's tell you about some of the sightings that have been seen. So this is since Christmas. Some firecrests, I don't know any numbers here, I'm afraid. Firecrests at Streatley and Wokingham. Hawfinch at Coombe, Aldworth and Mortimer. Bitterns, there are 1 or 2 bitterns at Dinton. In fact, I believe, my friend told me there might be 3. Merlin at Coombe and Lambourne. Scaup at Burnfield. Hen Harrier at Lambourne. Great White Egret at Moor Green, Padworth and Beale Park. Ringneck Duck at Moor Green and Fleet Hill Farm. Red-crested Pochard, 17, there's a figure there, at Hose Hill. Kittiwake at Padworth Lane, the floods. Siberian Chiffchaff at Lower Farm. But sadly, Julia, no house sparrows. Does anybody else have any sightings that they've had in their garden? Anything different? I haven't, but still no chaffinches, still no greenfinches. Yes, Sally.
I was going to say this, I get house sparrows in my garden every day. So I've got a small flock that live in my pyrocantha.
Oh,
so.
Oh, how lovely.
Golden eye at Dinton.
Oh, there's a golden eye at Dinton, I'm told.
At least 2 males and 2 females.
2 males and 2 female golden eye at Dinton. On Black Swan Lake, is that? Okay. Anybody else? No? Okay. Right. In that case then, I will now introduce Dr. Julia Schroeder, who is now going to tell us all about her trips, I'm sure, and the house sparrows on Lundy. So can we all welcome Dr. Schroeder?
Thank you very much, Sue, for inviting me, and thank you all for coming and for being interested in this. I hope I can, well, entertain you for an evening, and I hope it's not too academic. And I would really like to ask you if you find something too complex, or please just raise your hand or interrupt me and ask a question. Yeah, so I'm going to tell you about all the secrets of the house sparrows. We're studying these on Lundy Island. And to do that, of course, we go to that island often. And I've just spoken to one of your members.
Sorry, Julia, just to say that the camera for you is on here.
Yeah.
So if you can just there, then people at home can see you as well as the screen. Sorry to interrupt.
Yeah, I moved on. Yeah, so we go there by boat in the summer, and in the winter we fly by helicopter. And people come to me and say, why? It's quite an effort to go if you go by helicopter. You can just go, yeah, to your garden and have a look at the house sparrows there. Why don't you do that? So why do I study house sparrows on an island? And you're all ornithologists, so you know the main difficulty when we're studying birds is they just keep flying away. So they're very, well, there's many of them around and they're easy to observe, but the moment you think, oh, now I can really see that eye ring flying away, or diving or whatnot, and you just think, oh. And that makes it really difficult to study birds over a longer amount of time if you want to recapture them, if you want to follow the same individuals over and over again, not just in a sort of moments, snap sort of shot. Then it's really difficult, and then they're not very good study subjects. However, if you work on an island population, that is a bit like a natural laboratory, because house sparrows, they don't fly very far. Their dispersal distances from their home place are like a couple 100 meters. Some of them make it 1 kilometer to the next farm, you know, most of them don't. So, and they're really not well suited to long distance flying over large amounts of waters. And in that sense, when we study them on Lundy, we are able to catch every sparrow that lives there, like all of them. We are able to monitor all breeding attempts in a year. We're able to ring all birds. And if you go to Lundy and you look at house sparrows, they're all ringed. They all have unique color rings. If you find one that is not ringed, you need to tell me, because then I get a beer from my PhD student whose job it is to make sure they're all ringed. It's in fact on Lundy, so this is on the island. As you can see, it's a vast expanse of it's practically a rock with a bit of, you know, unimproved meadow, sheep. Then there's some soy sheep, there's some wild ponies, a lot of heather, gorse. And there's a small village where the red circle is, and the sparrows really don't go further than that red circle. Some of them in the summer, sometimes you see them at this lighthouse, which is in front here. So sometimes they go there to forage, but it's very rare that we see them there. So it's a really small place, and they breed inside barns. And we can put up mist nets inside barns, so we can really catch all of them, not only during breeding, but also in the winter. And that allows us to have a precise knowledge about their exact birth dates, because I know exactly in which nest box each bird was born. I know who their parents are. I know how old it is when I recapture it. I also know that when I don't capture it back, that is fairly likely dead. So I know fairly good their exact ages whenever I catch them. I also know their relationships. I know who are their parents, who are their offspring, who are their siblings, who are their aunts, uncles, and that sort of thing. Because they all have colorings, and when they breed, we put little cameras up and we just see who are the parents, and we know who is there, who's alive, who's not. But we also know extramartial relations, because we take blood samples of every bird when they just hatch, but also a bit later. Every time we recapture them, once a year, we take blood samples, sometimes even more often. So then we can look at genetic relationships. And because we take these repeated samples throughout their lives, that allows us to really follow them very closely and make, well, learn what happens as they age. And that is what we call longitudinal data, and that is really, really rare in wild populations, and it's even more difficult to get in bird populations because, as you know, there's a lot of dispersal going on in open populations. And that is what we call the Lundy Sparrow Project. We have a little logo also, and that goes on since Well, it started in the 80s when Terry Burke, who was my mentor, and actually the person who now runs the place that came up with eBird. But he was a lecturer and a reader at Imperial College, where I am now, and then he was Ian Owens, and then he was the scientific director of the Natural History Museum, and now he moved over to the US and just his things there. Anyways, when he was a postdoc, they did a couple of studies there, and then in the 90s people did a couple of studies. And from 2000 onwards, we started to do this in a systematic manner, and every year we go in the summer, we go in the winter, we capture these birds and do all these things. It's just to remind you, it's a short, house sparrows are short-lived passerines. They live up to, well, on average 3 years, but some can live up to 10, 12 years even. They're predominantly monogamous in behavior, at least, in social behavior. They do biparental care, where both parents care for the young. And they have 2 to 4 broods of about 4 eggs per year. So, as I said, we study them comprehensively throughout the year. We look at dominance hierarchies, we look at parental care, we look at their mating behavior and how they grow up. We study them in nest boxes. Lundy is about 20 kilometers or 15 miles away from the next mainland. So the only way they could go off the island is by being blown off. And we've actually done some surveys across the Devon coast. And we also looked at genetics, and some of them are related. Actually, I think they're blown off the island. I don't think they could, because it's northeasterly wind and northwesterly winds that blows them there. Sometimes we also have incoming birds. I think they come by boat. You know, in the summer, when you go from Ilfracombe or Bideford, there's a nice harbour, and there's people, they sit on the boat, they eat. There's lots of sparrows in Ilfracombe. I think they just hang out on the boat, maybe pick up some of the cake, you know, and then they find themselves in the open seas and they think, well, this is it now. So it's very rare. In the last 20 years, we had 4 immigrants that are confirmed genetically. Yeah, we check the nest boxes in summer. Actually, this is my son who's sitting there now. It's a bit the one who's looking into the nest box on the right-hand side, on the top of the ladder, a bit taller now. In the winter, we catch them by misnet to just see who's there and what is survival over the winter. Obviously, we ring them, but most of my captures are actually not ringing captures because they're not ring, you know. So we measure them obviously every time, and it's a bit of a total surveillance. And if you look at this picture, you will see that all of them have colorings actually, and they're all individually coloring. They also have RFID tags. So we tag them, they get a little tag implanted under the skin. And then when some of our nest boxes are equipped with RFID readers, when they go in and it goes beep, 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 and it registers the individual bird, which is quite nice. As I said before, we take blood samples. I have a license from the Home Office to take blood samples of these birds. It's not a lot, it's really just a little drop. But with this drop, we can, as I said before, we can determine their paternity. And we do this by microsatellites. And this is just a quick example of how it works. So if the mother is homozygous, that means she has 2 copies of the same allele. In this case, it's 100. And then we also type the offspring, and the offspring is heterozygous, so it has 2 different copies. It has 100 and 104. And so clearly it has the 100 copy from the mother, because the mother can only give you 100 and nothing else. And then if we type the social data and we find the social data has nothing in common with the offspring, we know it must be an extra pair offspring, right? And this is where most studies stop. They only know that there is an extra pair offspring because they aren't able to monitor all the males, you see. But because we have all males, we are usually able to find the father and say, oh, this male B in this case must be the father because this one has a copy of the 104 allele. And of course, we do this not only with one marker, but well, by now I think we have 17 markers. We genotype more than once, and we also sample them more than that, and that gives us very high confidence in our paternity assignments, and and that gives us quite some insight into the secret lives of the sparrows. So by now we have about short of 20,000 DNA samples, and that's on average 1.5 per bird per year. And that allowed me to put together a multi-generational pedigree. And this is what it looks like, so it's quite massive. You don't need to understand it. It's the red lines are maternal lines from a mother to an offspring, and the yellow ones are paternal lines. But why I'm showing you this is it's really unusual to have that in open populations. You usually have like these sort of unknowns hanging out there. And that's just an overview of our data. So here in the graph, you see on the y-axis is the number of breeding adults per year on the x-axis, and you can see it's quite fluctuating. And there's a couple of interesting things. When we started in the 1990s, there was a big effort going on Lundy to eradicate rats, because I had rats, and the Manx shearwaters and the puffins were in decline. The puffins were completely disappeared from the island because the rats would just eat their eggs and their chicks. And so they flew in experts from New Zealand who tried to get rid of all the rats. And that's been very successful. By 2006, the island has been declared rat-free, and it was the first time that this has been done outside New Zealand. But they started off using rat poisoning that is poisonous to birds, and then all the sparrows started dying, and we convinced them to change the other point, and that was successful. But we also brought over a couple of birds. But as you can see, there's quite some fluctuations going on. People sometimes ask me, why is that? I like to say it depends on how many chickens they have on the island, because that is correlated with how much they feed the chickens. And that's what the sparrows like to feed on. We had a bit of a dip now. I think the population has been crashed last winter, because this only goes to 2019. We've been there in 2020 and 2021 despite COVID, but this year, this winter, it was quite small, and I think it may have to do with that they had to get rid of the chickens because of avian influenza. So it's the jury's still out there. But anyways, what I want to show you is we have on average about 60 breeding pairs per year. We have over 2,700 groups by now of 11,000 birds, and we have a lot of information on all of them. Not only do we have the social pedigree and the genetic pedigree, but we also have the foster pedigree. We cross-foster birds, where we take them from one nest, put them in the other just after they're hatched, and that allows us to look at things like what is genetic and what is environmentally influenced, what kind of traits. We also, and I will talk a bit more about that later. We know about parental care, about social associations, about their personality, about who's dominant bird and who's not, and all sorts of other things. And that allows us, of course, to answer quite interesting questions. But the most important part, why I'm doing all this and why I'm going to this great length trying to explain this to you, is that we get relatively precise fitness estimates for wild populations. And that brings me into my main topic. So I'm an evolutionary biologist. So I'm not going to talk to you about conservation or bird watching, but I'm going to talk to you about evolution. And that brings me to Charles Darwin and fitness. And fitness is actually quite important. It's a concept that is important for evolution, and that is called or defined as the average contribution to the gene pool of the next generation. And that the idea here is that genes can, well, propagate and distribute and be successful if they are being distributed into offspring and their offspring and so forth. And so what is important is that we measure that somehow in wild populations. And if you have like a couple of sparrows that breed, have 4 offspring, then these have offspring themselves again. The problem is, how do you measure fitness? Where do you start and where do you stop, right? What is fitness? Is it the number of eggs? Is it the number of hatchlings? Is it the number of fledglings? How do you even measure this when you go out and do that, right? What is a good proxy for this sort of elusive fitness that you can measure throughout generations if you want to? And what we've done is, and because typically you can't do this because we don't know, but on Lundy we know. I know exactly which birds have survived and which offspring have survived, and I can even look at which genes have survived over the last 20 years. And that is incredibly rare that you know this. So what we've done, Shiva has done that, she's a master's student. She's looked at the correlation between this actual fitness and the measurements that we usually take in the field, like the number of eggs, fledglings, hatchlings, or even recruits, and how good that correlation is with actual fitness. And what she found is that only the number of recruits is highly significantly correlated with actual fitness, while, so this is the correlation on this x-axis. And if this bar overlaps 0, that means there's no correlation with fitness. And only the number of recruits and the delight fitness, which is a compound that is calculated by the number of recruits, is actually indicative of fitness. And that means when you go out and have a population where you don't know what happens to the offspring, these values, how many eggs, fledglings, and hatchlings, is actually not really informative if you have evolutionary questions. And you actually need to see how many of these survive until the next year and breed themselves. That is the definition of recruits. So yeah, I would like to drive that home. We have very precise estimates, and therefore, and that is unusual, and therefore we can answer evolutionary questions. Yes?
What do you mean by taking a bit?
Oh, I'm sorry. Recruits is, yeah, it's good you're asking. It's a jargon term in evolution, and recruits are the individuals that survive themselves and breed themselves.
Okay.
So in my case, these are really birds that are born, live for a good year, a bit less than a year, breed themselves and have offspring that we also genetically measure that are there. So that is quite a compound measure, and to get that in an open population is often near impossible because they just fly away and reproduce the wilds. And as such, the Lundy Sparrow project is very unusual. It allows for answering evolutionary questions. And in this talk, I want to talk to you about some of these questions that we can't answer because of that. And one is, why do birds cheat on their social partner? What do social networks have to do with that? And then I've been told there's a break. So after that break, I would like to talk about aging and why age, and about transgenerational effects of age, which is quite fashionable these days in where I work. Yeah. So when we talk about why birds cheat, the first thing we need to talk about is anisogamy. That is a mouthful, and what that really means is that the gametes, or the cells with which we reproduce, are of different size in males and females. So here you have an egg cell, a human egg cell, and a lot of sperm cells. And I think this picture really drives home that females have fewer gametes, that's the word for these cells, and they're bigger, while males have many more, near unlimited supply of them, and they're really small. And that is this very fundamental difference between male and female, between the sexes in biology. And that has massive consequences. And I just looked that up because I was curious. I looked at how many children can women have, humans. And when you do that, you find this lady who is a Russian lady, and her name is not known when I looked that up, but her husband's name is known, but I won't tell you because I think this is about her, nothing. And she has 69 children, and 67 survived infancy, and they're all in the picture. And according to Wikipedia, at least, this is the woman who has most children, and she had obviously a lot of twins and triplets and whatnot, but I still think that is an extreme amount, and just and all of you kind of breathe in, and I think that shows what kind of energy goes in that, right? And how many resources this woman must have depleted by going through this ordeal. If we go to men, we have this Ismail Ben Sharif. He had a harem, and supposedly he had over 700 children. And then we have a bit more recent, in the 1700s. This is Berthold Wiesner in the last century, and he had 1,200 children. He was a fertility doctor in Switzerland. But I mean, 69, and let's say, well, 700, it's about more than a tenfold difference. And none of you went like, oh, when I said 700, because for a man, it's not that much of an effort, honestly, right? Yeah, but that is a fundamental difference in men and women, that women invest much more into smaller amounts of offspring, and probably care more about quality, while men have this quantity thing going on. So there's this trade-off, and there's this sort of sexual conflict going on, where if we talk about cheating or, you know, multiple, well, it's impossible for women to have more than one child at a time, other than if it's twins, but of more than one man. It's practically impossible because you just have one womb, right? If you can't have another child at the side, well, that is easy for men to do, right? And if men do that, they don't have to raise that extra child necessarily, right? Because they're kind of, there's another effort that is not included. Women have to raise their children. There's no way for women to escape it. So there's not just the pure biology of it, but also the consequences afterwards are mostly borne by women. And those that have their offspring with an extra pair male often bear even more of that. And this is what I want to talk about, because we also see that in birds. And in sparrows, actually, they often have 4-ish chicks, like a pair. They often stay together not only in a year, but even the year after. They even sleep in the same nest boxes in the winter next to each other. We know that. But if you look at the genetics, you will often find that one or even two of the brood are by a different father. And when you look at that over the years, we find that about 24 to 49% of the chicks in our house sparrows are extra pair, have a father different than their social father. And the question is really, why do females do this? Because we just went through all of this. Females got, you know, a sparrow female, she can lay her 4 eggs, but she can't go and lay a 5th, a 6th, a 7th, and 8th. But a male can do that. He can just go, I don't know, to have 20 offspring easily, right? So there's a clear benefit to males, supposedly, but what's the benefit to females? And there's a lot of theory about that in evolution. And the first assumption is that males actually gain this direct benefits. Males that cheat, we would expect them to have a higher fitness than males that don't cheat. It's a simple thing to test. The second one, and I'll talk about that a bit more, is that females pay costs. It's costly for females to do that. But if they do so, we need to find some benefits. And these benefits are elusive, and we're going to hunt them down in this talk. But first, I want to talk about these benefits that males supposedly have. The idea is males that cheat or are not monogamous have more offspring. And what I've done with our Lundy data is I've divided males into those who cheated on their partner, we call them extra pair males, across their whole life. This is. We took those that remained monogamous, and I can do that because I have data of all offspring. So usually you can't do that because you don't know if he's just gone around the corner, you know, in the next park and sired an offspring. But I know that because we know there's no other nest. And then we have something that we call floaters. These are males that don't have a social partner, never had, but sometimes they manage to sneak in paternity. So the extra pair ones are the ones that actually have a social partner, but also sired offspring elsewhere. But when you look at how many lifetime offspring they produce, that is the y-axis, and you see that there's an average of about 6 lifetime offspring that birds produce. You will find that the extra pair males produce most, the monogamous males produce second most, and the floaters produce least number of offspring. So what we can say definitely is that there's an advantage to cheating, and that also transports down to the number of recruits. Remember, I've told you the number of recruits is more important, but that still remains the same, and even though on average there's one recruit per individual, those that cheat have more. So there's a fitness advantage, an evolutionary advantage to these males. So we can say, yes, this is indeed true. It's a bit more difficult. We think females pay costs of this because we think, and that's a theory, that males retaliate. When a male knows she's cheated, he's got a problem because he's got to care for offspring that are not his, right? So typically we have costs and benefits of raising young. So if you raise your young, you put in all the parental care, and if you have had children and got up at 3 o'clock in the morning to work, you know, I'll pay for it, right? But you only benefit from that when it's your genetic offspring in an evolutionary sense, right? But the costs, of course, occur independent of the relatedness. So if a male has 4 chicks and they're all related to him, the equation turns out to be fine. He pays for 4 chicks, but he also gets the fitness benefits. But if one of them is not his, he works too hard, really. So evolutionary speaking, he should stop and he should care less. He should reduce his parental care. And it's been a very, he should reduce his investment. And that's a very popular research question. Do male birds adjust their care to the amount of extra pair young in a brood? And with the advent of molecular biology, people have gone out and done that, like often, and they've just plotted parental care on the y-axis, extra pair in the nest on the x-axis, and they were hoping that there would be less male care in the brood. So people have done that so often that people have done a meta-analysis. I don't know if you know what meta-analysis is, but this is an analysis of all those studies who did the same thing, and then they look what the average effect is. And here, this Z adjusts the average effect, and here you have the species that has been done in, and that's not only birds. I hope that's enough. And you see that there's, in general, there's an effect, and they say, oh yeah, there's an effect. And males do adjust, but I said there's problems with this analysis because they did not follow individual birds. They just looked at individuals, and what they found is that those males that probably are of less good quality, are maybe exhausted or sick or whatever, do not provide a lot of care, but maybe the female also thought, oh, he's not so good, maybe I'll go find, you know, so it's not really a good test of the hypothesis because it compares between males and does not follow individual males. So what you really want to do is you want to do it within the individual versus cross-sectional analysis. So you don't want to compare a crappy individual to a healthy individual. If you would do that, it's easy to find that relationship. What you want to do is you follow them. You want to look at a healthy individual getting skinnier and see what happens then. Does it reduce its extra pair and also its care? Or does the female increase the extra pair, or does it stay on the line, right? When the female changes her extra pair. But it's really difficult to convince the female to change her behavior, you know? So you can't really do an experiment. That is really difficult. So what you need to do is you need to have repeated observations on the same bird. And very many data points, which is what we have. When you look back at that, you'll find that about 6 analyses in here had repeated measures, and the most I had was about 10 individuals. So there's really poor evidence for this. So I thought we'll use the Lundy data to the rescue. When we have more than about 200 repeated measures on the same individuals, as well as provisioning visits and incubation time, which is quite a massive data set, I think. And then I did, and I'm not going to bore you with this, some statistic. This is interesting if you like statistics, or if any of you is interested in that, come talk to me. I really like that. But you can actually disentangle these effects from within individuals, and that is what I've done. And I found that males do not adjust their incubation time when their females cheated. But when you look at provisioning behavior, the picture changed, and what you find is, and this is the within individual effects, you find that they actually do reduce. So they provide less care, they feed the young less often if the female has cheated on them, and it's a within individual effect. But that only occurred when they changed the female. when they stayed with the same female and the female changed her behavior, the male always provided consistently the same care. While if the female died or he chose a different partner, then he changed his care into the direction depending on what kind of level of extra care she would seek. And females were also quite consistent in whether they would cheat or not. That was quite interesting. But however, for us, this means that, yes, there is a cost to female cheating, but we also found that they have kind of consistency, that females cheat once, will cheat another time, and will have males who have lower care. So, but this, of course, puts a problem, because if it's beneficial for males, but if it's costly for females, then evolutionary, why do females do this? They should just stop, right? Because it's not good for them. So the question is really, why do females cheat? And evolutionary biologists think there are some benefits that are less clear, less obvious, more indirect. And these benefits are genetic diversity, compatibility, and good genes. And I will talk you through this, hopefully. So the idea of these indirect benefits is that if they cheat, that first of all, the one they cheat with, the male, the extra pair male, might be of higher quality. So if they are with a male and they think the quality is not good enough, then they could go and seek extra pair populations from a better male that has better genes, because he does not provide care or anything. So it's only the genes that get. So we can test for that, right? We can look at whether the male is better than the female, than the extra pair male is better than the within pair male. But it also predicts that the extra pair offspring are better than the within pair offspring, because they have better genes in that case. And these are the indirect benefits we wanted to look at. So these are the two predictions: extra pair males are better than within pair males, and extra pair offspring are better than within pair offspring. And these better can be anything in terms of fitness, in terms of size, body mass, but also in terms of genetic diversity. We looked at this, and this is an example with the expected probability to hatch. And here what we are comparing on the x-axis is extra pair offspring, the probability to hatch, with within pair offspring of a brood where there's extra pair in. And then we compare them with within pair offspring of a monogamous brood. Because clearly in that case, the female thought, oh, I have a good male and a female. So you need to kind of distinguish between these. But to cut a long story short, we did not find any differences. And that was also true when we looked at nestling survival, at recruitment, and lifetime reproductive output, and any sort of fitness measurement. We did not find any benefits for the female. And then we thought, well, maybe the male is still better, we just don't really figure it out. But here also we did not find, and it's a similar figure as I've shown you before, if the bar goes to 0, there's no effect. So we did not find any differences between these males for whatever genetic similarities indices you pick, body size, ornaments. The only difference we found is that these extra pair males were older, which is kind of not a benefit, you know. So we don't know. So when we go back there to our theory, it's like a big no. We needed to go back to the drawing board and read back on what Darwin said and what other people said, evolutionary theorists said, and what could be the reason for this, because this is basic sexual selection. It's really basic theory, and we struggle answering this. And the last slide I showed you actually was not only Lundy Sparrow data, but it was data from all studies that ever worked on this before. It's a meta-analysis in birds. So in birds, females that cheat do not find any indirect benefit. So it's a very general finding. Well, then if you go back to theory and you find something that is called non-adaptive theories, and these non-adaptive theories are still a bit vague, they're less clear, but it's something that we can look at. The first one is that same genes drive the behavior. So if the gene that makes a male cheat, and he produces daughters that have that same gene, they might also cheat, even if it's not good enough for them. But if the benefits for the male outweigh the benefits for the female, maybe this behavior can still evolve. And in this case, you would expect that males that cheat will produce daughters that cheat. That is a premise that is really difficult to test, because you need a ton of data and the quantitative genetics, and we've been doing this since 2012, and we think we don't find that, but it's difficult to say because even with our data, we don't have the statistical power to do that. And the other is that females that have a high libido within pair, which might be good because that might mean they have a lot of offspring, but they might also be more likely to cheat because it kind of spills over. And to look at that, we didn't look at Lundy sparrows because you cannot look at copulations in the wild. That is really difficult, as you know. Sometimes you see it, but you not only want to see it, but you also want to read the colorings, you know, of both. And that is really difficult. So I have this colony of house sparrows captive, where I do experiments on. I don't do any mean experiments on them. I don't kind of poke them or anything. We just do observations. They, as you know, they like to breed in a sort of barns and stables, so they're really easy to keep because they like this sort of setup and they're very sociable. And when you stand there with the binoculars for long enough, you can see all these populations, and that is really nice. I have a video that is explicit. Are you okay to watch that? Let me see. Can I? Is it also on the Zoom?
It's not playing, unfortunately.
No, but you can see it. Yeah,
Is it,
it's very short. Oh, yeah. Wait. And this is something that we videoed. So we take videos of these nest boxes. This is not. I should have remembered. Oh, there's the female coming out of the nest box. You've seen the rings, right? Oh, you don't? It's here.
no. Oh,
Maybe.
I
I can show it to you later, maybe.
No, that's all.
Hmm.
Oh, I see. No, I see you linked to a tweet. Oh, no, I know.
Yeah. I'll
The trouble is, it's not on the.
Oh,
it's
you
not
know
on
what?
the. It's not on the screen. You need.
No.
It's on YouTube, is it?
Yeah.
Okay. The only way we can do it is to move that across to that screen, which is the one that's being.
Is that trouble?
I'll have a go. It's the only problem I get. Oops, not one. Is that it's.
Just such a nice.
I can never remember which way it
It's
is.
in
There
the
we go.
same. Here.
No, it's the Zoom. Everybody here and Zoom can only see what's on the big screen. So I'm hoping. Is that on my Zoom screen? Okay. If I could just. I can't. Yeah, I can't. Sorry, I can't see it because. Sorry.
Maybe colorings. This is nice. There. Did you see that? And it's awesome because you could see the colorings, you know, of both of them. Look at that. And we never. This is really unusual, and we have like a couple thousand videos, but only one thing. So only when they actually touch is the ejaculate transferred. And the female kind of invites him in when they're sort of lifting the tail. Yeah, shall we move?
Okay.
No, it's on the move. Shouldn't we? That's not on the move.
Show it to the end or the.
Yeah, well, it goes on.
Yeah, right, fine. I'll leave that in the right place in the room.
How do we go back?
Well, I'll just need to we stop that first. Have we had enough sparrows?
It's really unusual, you know.
I
This is
think
a
I
flick
should
of one.
be able
We're
to.
really keen.
I'm not sure
Why
we're going to be in
having
the same
offspring?
place.
You know, eggs or chicks in the nest box, otherwise we wouldn't take a video. So the other day they have up to 3-4 broods a year.
Oh, the only trouble is we're not in the right place now. Do you know what? You sure? You need to zip across to. Do you want me to stop it and move on, go back?
No, this is fast.
You sure?
Yeah.
I can. Would you like me to?
Yeah.
This one?
Well, yeah, it's stuck a bit, is it?
No, let me.
Yeah, I think maybe not. Well, no, maybe.
Oh, okay. Oh, no, do you know which slide?
Yeah, a bit.
No, I'm struggling to see it because it was.
Yeah.
How far down are we?
A bit down. A bit more. A bit more. Oh, yeah, if you go there, I think from here.
Yes, it is.
There.
That one?
Yeah, yeah.
That one or the one
The
before?
one before, but I can just move up then.
No good with Bob's mouse.
If you go here.
That one?
Yeah.
There we are. Hopefully, you're back to now where you were.
So we've been, my student has been very proud of that video. So, but what we've done is we've done that indoors in the aviary, and we've counted them. And what you actually looked at is the number of total populations of female would do on the y-axis, which went from 0 to 20, and she correlated that with the number of extra pair offspring. And she did not find that sort of behavior that we wanted to see, unfortunately. So here we also have to put a little X there, and then what was left in our arsenal of hypotheses was really, maybe it's just chance. You know, maybe females just do it because there's an opportunity. And when I say opportunity, you need to have sort of like a dance to flirt, and you need to have a male, right? If you're just sitting alone at home in a sort of lockdown situation, there's no chance of flirting or, you know, anything happening. So what we think is that if we look at the social network of the sparrows, that we can maybe find out if she has more connections, if she knows more males, maybe then there's more opportunity and she might be more likely to engage in extra pair copulations. So the idea is that it's a spillover behavior, mostly driven by chance and by the network. And we actually have information about the social network of house sparrows, because I've told you before they have this RFID tag, and we have this little black rectangular thing where a cable comes from is an antenna. And we have these feeding stations, and we put antenna around, and every time a sparrow lands on that feeding station, it goes beep, 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 beep. And with this, you gather a huge amount of data in like a very short amount of time. And then you know which individuals hang out together at this feeder. And then you have to kind of come up with a definition. What is a friendship? What is a social association? Here you can see, for instance, under A, you have 5 birds that have been detected, that are RFID tags, that there's also some of them stay for a time at the feeder, then they fly away, then they come back. So these bars indicate how long birds stay at the feeder. And the question is, what kind of overlap do you then define as friendship? And I have a PhD student who spent 3 years of his life on that question. And he's come up with something where he said, well, if they landed together, and then spend some time there, they're likely associated, right? And I'm just showing you this to tell you that we spend quite a lot of time on that, because it's different for different birds. If you have, for instance, great tits, they have a different behavior in summer and winter. They have different social behaviors, so you need to take these things into account when you want to build these networks. But Jenny's built these networks, and what you can then do with these networks is you can count things. For instance, and we call that degree, which is, again, jargony, but the degree is how many friends you've got. So when you go to Facebook, you've got a number of friends, that's the degree in network speech. And we can count how many connections a sparrow has. But then you can also calculate something that's called centrality, and that is related to how central you are in a network, with how many are you connected and how short Do you know the 6th degree of separation? Well, the idea is that you know, for 6 people, you know, I know Bill Gates, something like that. And that is this sort of centrality idea of how many connections you have to get to a certain person. And you can calculate that, but I won't bore you with that. And then there's also strength, which is the number of friends, but weighted by how often you meet with them. You know, so if you meet one person more often, then, or most of them more often, then it gives the whole thing a bit more weight. And that gives us a number of variables to play with that kind of tell you something about extroversion and introversion of the sparrows and how sociable they are. We've got quite a lot of data, like up to 200 data points for males and females. More individuals have fewer data points, so there's quite a large number of individuals that have more than 20 data points. So we are quite sure that we have good data here. And what we found is this table, what it tells you, the repeatability of these traits, that sparrows are incredibly consistent in these behaviors. And that is true across time, but also across seasons, so winter, summer, but also in captivity and on Lundy. So we find the same things in captivity and on Lundy, and that means this is really a biologically general observation that we've made. Sparrows are consistent in how many friends they have and how extrovert and introvert they are. And we can find that birds that cheat are slightly more extrovert, which is quite nice. We also find that the more extrovert birds have more partners, be it social partners or genetic partners. When you sum them all up, you find they have more. So there's something to this opportunity theory, but we were still not too happy with this because we thought it's just all observation. Can't we do an experiment? You know, we're all scientists. And what we've done is we have these aviaries. We went back to the aviaries and we measured their personality, and then we set up aviaries. One aviary where we had extrovert males and extrovert females, one with extrovert males and introvert females, one where that was the other way around, and one where there was only introverts. And we predicted that those that had most extrovert females would have highest extra pair afterwards. So we let the breeding season run its course, and then later on counted what happened. And what we found is, and high is high extrovert, H and L is for low, and the first one is the male and the second is the female. And what we indeed found is that the ones where there's only extroverts, so they had an orgy, you know, while the introverts didn't so much. So there was an effect, and we found that whenever there were extrovert birds, there was more extra capacity than when there were none, which was quite nice because it's an experiment, it's a manipulation, and it shows it works somehow. So maybe opportunity plays a role, and we're still working on this, and we're looking at that in the data, but I don't have the data yet. So why do females cheat? Even though males retaliate, and even though males reap more benefits than females, why do females cheat? We think it might be a result of opportunity and maybe some genetic correlations. And that was the first part of my talk. Do you have any questions? Yeah.
How, suppose you have a nest with 4 eggs in it,
Yeah.
how does the male know which one's not his?
That is a really good question. We don't know. And we think they don't. So we
How did they end up there?
yeah, we've done tests because we also do cross fostering where we swap chicks, and then you can see whether males would change their behavior depending on that, and they don't. I think males don't look at the chicks. I think males look at the female. And we also, what we did not find is that males did not react to the female's behavior. They reacted to the female's identity. And if they would change their partner and have a female that has a lower or higher extra pair rate, they would change their behavior in the respective direction. But so what we think is that there's some sort of personality trait of that female that kind of gives it away, maybe, or maybe there's a other not measured variable that correlates with both. Yeah, it's possible. Yeah.
Well, who knew? Yeah, that was perfect, thank you. We're now going to have a 10-minute break for some refreshments here. So we hope to see all of you at home back here in about 10 minutes. Right, we'll have the second half of our meeting now, and I can't wait to hear what's going to happen.
The inevitable. So I would like to talk about aging now, so we're changing tack quite a bit. And as I've said before, because we have this data on sparrows, we can learn about how wild individuals, how wild populations age. And aging, evolutionarily speaking, is actually really odd. So when it comes to evolution, we shouldn't be aging, because any organism that would evolve a gene or a locus or any mechanism to overcome aging would produce so many offspring constantly that it had the biggest fitness of all, and it would take over the planet. We had no diversity anymore. We just had this one Darwinian demon, is what we call that. But in this thought experiment, why didn't that happen? Because evolution has overcome so many obstacles, but this not. And that is really, really odd, because it's this fundamental constraint and limit of life. Yet we find senescence, aging, it's everywhere in nature. And we have a number of longitudinal studies where people have followed individuals across their life in the wild that have found that wild birds, wild animals age. People thought this wasn't the case. People thought they would die before aging sets in, but that is not true. And even in the albatrosses, we can find that. But there's a large number of animals, and not only birds.
Excuse me.
Yes?
Can I just ask you what you mean by aging? Because I think maybe it means different things to different people.
So if I'm to explain what I mean by aging or senescence, it is the decline over time, over age. As we age, that some of our body functions decline and are less good than they used to be. When we go in the wild, we measure that. We usually look at reproductive output, and we see that animals produce less and less and then fewer offspring throughout their lives as they age. And that has been found in many of these species. But you can also find that in other traits, for instance, body mass, or how well they provide parental care, and these sort of things. So there's a lot of traits or health declines. And you find that in the wild. So it's not that these animals die before senescence, aging sets in. It is actually that they live long enough that we can measure that. And we've also found that in the sparrow, of course, but I'm going to tell you a bit more about that. When we talk about aging, we of course also talk about mortality and dying and the risk of dying. And typically, so I'm going to give you a bit of theory again here. Sorry, that's what happens when you invite a lecturer. There's two components really when we think of aging and death. And one is extrinsic mortality, and that is not dependent on age. That is catastrophic mortality, where just, thunderstorm, you're struck by lightning and you're dead. Can happen to anyone independent of age. It's just a catastrophic event. And that's one sort of mortality, and that's called extrinsic because it comes from the outside. Well, there's also the intrinsic mortality, and that is age dependent. And this mortality, the chance of dying, increases as we age. And if you plot that, right, you look at the intrinsic mortality, the chance of dying because of biological reasons inside you, is quite high when you're just born, when you're 0 years old. This is age on the x-axis, on the y-axis is that chance of dying. But then after infancy, it drops to fairly low numbers pretty soon, and we don't have in middle age high chance of dying. But then as you get older, the chances of dying increase again. While the extrinsic mortality is the same throughout your life, right? Because you can always fall downstairs or be hit by lightning. And this interplay of varying chances of dying over lifetime is what we think, evolutionary biologists think, the reason for the fact that we age and we have to age. And the theory is that natural selection is much stronger early in life than later. And the reason for that is, if mortality kills a baby, that baby will never ever reproduce. Never. Any genes that are in a young individual that dies early before it has children of its own also go extinct. Boom, gone. While in individuals that have already reproduced, that already have produced children, if they die, evolutionarily speaking, it doesn't matter that much, you see, because their genes are already propagated. I don't know if any of you has read Richard Dawkins' selfish gene, but once the genes are there, it doesn't matter, the individual doesn't matter that much anymore. So there's not that strong selection in later years. And the idea is that in later years, we might have genes that produce an illness, a disease later on, and they're not pruned because we can reproduce before, and these offspring have the same genes, and they reproduce, and these offspring have the same genes, maybe before that illness even actually comes to place. And therefore, these sort of genes that produce mortality at later age persist, evolutionary speaking. And another reason for this is there's more babies than older people. So there's much more selection going on early on. There's much more stuff happening. Well, at later age, there's very few left. And then they cannot contribute that much anymore to selection. And the idea here is also that you have limited energy, and that is called the disposable soma theory. So there's one theory about aging. The disposable soma theory also has to do with this. And the disposable soma theory, and I will explain to you why my dog and my chicken are talking for this. So please don't be distracted. The disposable soma theory says that maybe we just have a limited energy, and we need to make a decision whether we want to spend that energy early in life or later in life. And if we spend it early on and reproduce and have offspring, that's a safe bet. While if we wait until we get older, we might just be struck by lightning and not be able to spend that energy, right? There's a bigger risk involved, and therefore natural selection favors early reproduction over late reproduction, and that is the idea. And I've tested this disposable soma theory in chicken, and I thought, since you're ornithologist, you will excuse me doing this detour. I will come back to sparrows afterwards. So yes, the idea is we have a limited amount of resources, and the idea is we live now, we can suffer later. It's better than the other way around, right? Because it's a safer bet. But we have very little evidence for this, actually. We have some evidence between species, such as elephants live really long and they reproduce very little, while insects, for instance, have these high reproductive bouts very early in life and then die right away. But that is between species, but we have very little evidence within species. And there's some correlative, but we don't really have experiments. And the reason for that is you can't tell a bird, no, do you want to lay a few more eggs? You know, it's really hard to do. Or you can also not stop them from laying eggs. It's really difficult to do manipulatively. So this is why, yeah, we don't have that. And there's limited experimental evidence within species. So what people do is they do clutch size manipulation. They give birds more eggs, and then I have to put in more parental care. But that comes with other limitations. So we really don't know what's happening. In 2007, I did this really interesting analysis on dogs, not birds, sorry. But what they looked is they looked at the average lifespan on the y-axis and the average weight on the x-axis. And what they found is that dogs, I was about to say birds, dogs that are heavier live shorter, and dogs that are really small live longest. And the idea here is, again, this was the soma theory. Maybe if they have more energy to spend, they need to spend more in other. But it's really difficult to explain. Also, it's not reproduction. So body mass is not reproduction. It's not that they reproduce a lot just because they're heavy. So this seems to be like a rule, but we don't really have a good explanation for that. And I don't think it supports the disposable soma theory. And in come my chickens. Lockdown, I have all these children who say, why don't we get chickens? I thought, okay, you know. And I don't know if you know about this, so there's this whole thing about rescue hens in the UK, where you can just sign up and then do a donation and you get chickens for free. It's called Fresh Start for Hens or hen adoption. It's a whole thing. And the reason for that is that after these hens have been in laying, they're usually layers. They're quite spent after a year and they stop laying so frequently. I have this here. So what happens is after 16 weeks of age, x-axis is weeks of age of a hen, and y-axis is egg production. I don't know, it's egg production. More is better. And what you see is that, is the egg weight. But what you see is that as they hit week 17, they start laying eggs, and they lay 1 egg a day. And then as they age, as they get older than a year, that reduces, and they lay less than 1 egg a day. And in the industry, in the egg industry, it's not economical anymore. So what they do is they cull all these birds at a bit more than a year of age. So these charities come in and say, oh, we'll take all these chickens and we give them to people who want to have them in the backyard, which is what we did. And this is how they come in, and then you give them a bit of food and a bit of love in your backyard, and then they become really, really feisty. And they eat all your vegetables, and my yard doesn't look that green anymore. And they even come through your cat flap. And then they walk into your kitchen and they go like, can we have some food? And the dog is afraid of them at some point, and they run your house, pretty much. But they're funny and they're really interesting, and some of them die very early, and that made me really think, because they're spending so much energy in laying 1 egg a day. And if you look at that, the egg weighs 100 grams. What does a chicken weigh? 2 kilos, 2.5? Just imagine, in 3 weeks, that chicken has laid its own body weight in eggs. And that is, we spoke about that in terms of female reproduction. That is like a massive energy investment. And these chickens have to eat that. And then they have to produce these eggs. And of course, they're bred for that, right? If you go and find red jungle fowl somewhere in Indonesia, they're going to lay, I don't know, 3 clutches a year or something like that. Surely not an egg a day. But I just think there are really these sort of factories of high energy food. And it must be really exhausting to be a chicken and to have to do that. And I thought maybe we can test that sort of disposable soma theory there, because it's so stark, right? And then you have, of course, different breeds of chicken. And some chicken are not bred for egg production. Some chicken are bred for fighting. Actually, cockfight is the whole thing. It's what is really brutal. You don't want to know about that, but they are bred for different purposes, right? Others are bred for feathers or just for showing off or for fun even. And I thought, well, if this disposable soma is really a thing, then chicken that are bred for laying eggs should have a shorter longevity, life expectancy, than chicken that are bred for other purposes, right? So what I did is, there's different, they look so fun. Chicken are awesome. So I had this master's students, and I said, look, this is a really nice master's thesis. Why don't you just go and find me estimates of longevity of different chicken breeds? And he went down and he looked, you know, we're all scientists, we look into papers, peer-reviewed papers, and he found 6 estimates. And I was like, okay, it's not that many. And so there's not much published on that actually. So what you have to do is you have to go into the grey literature and on the web. And because people publish that sort of things, farmers publish that. There's a lot of, societies, it's a lot of amateurs who publish that, but it's not peer reviewed. And you find all these data when you crawl through that. And that's what he did. And he found 704 estimates from 18 sources and 180 estimates from 10 sources in the grey literature. So this student was really good at that, and he found like all this data. It was awesome. And so he found breed level estimates for 164 breeds, and he found the problem was you don't really get lifespan. So you often get average lifespan, but sometimes you don't, but then you get the lifespan they are productive. So how long they lay eggs or when they stop laying eggs. So we use that as a proxy, the productive lifespan. Of course, you also know how many eggs they lay. So the idea is chickens that lay an egg a day should live shorter than chickens that lay, I don't know, 2 eggs a week. And this is the result. This is what you found. On the x-axis, you have the annual egg production. And as you can see, some lay up to 300 eggs a year, others not. And then the longevity on the y-axis. And there's a significant effect. Indeed, that those that lay more eggs live shorter breeds. There's a breed average than those that don't. And this is interesting because these are only chickens that are bred for egg laying. And when you add all the other ones for down and meat and fighting and whatnot, it gets even stronger. And you can also see that all the others that are not bred for egg laying are actually to the left of this, and they don't have this extreme high output. And they also live all a bit longer. So I think if you select artificially for high reproduction, you also automatically, inadvertently select for shorter lifespans. And that is, again, suggestive of this really hard trade-off, that link that evolution cannot overcome, that we have to age. And it's supportive of the disposable soma hypothesis. And I really like this study, that's why I plugged it in. But now we go back to sparrows, like my chicken. Okay, let's have a look at sparrows and how sparrows age and what's happening there. First, I have to bore you again with a bit of statistics. Hope it's not too much. And I want to tell you why it's important that we measure the same bird over and over and over again, instead of comparing old birds with young birds. Say you want to test whether fitness, reproduction declines with age. And you make a plot like this, where on the y-axis you have fitness, number of eggs, or whatever. And on the x-axis you have age, and then you get this pattern. You can say, oh, maybe there's an increase, you know. But if you would look at that and look at which of these dots are the same bird, just measured across time, this might actually cloak any patterns. It might actually be that there's a decrease, but we don't see it because we don't know if we've measured the same bird twice. Right, so it's, and I think this really highlights why you need to do this sort of longitudinal data collection on islands. But it could also be the other way around, right? But if you don't know that, you will miss these sort of patterns. And I think this is again to make my case for why we need to have repeated measures on the same individuals if we want to study aging. And that is why it's really, really difficult in humans to study aging. It's really difficult because very few scientists are able to follow humans from infant to old age because we just don't live long enough. There's a couple of studies that actually do that, where scientists just move on. There's generations of scientists working on these sort of data. But you can imagine how difficult it is to set this up and to convince people to give you funding for that, because you need to have funding across generations. And that is really difficult. And that is why a lot of human data actually is cross-sectional. And that is problematic. And that is why it is actually really good. I'm not only doing this for fun, but that actually gives us important data. We can also do that in mice, in lab mice. But the problem with that is that they are highly bred, that they're bred, inbred. and they don't live under natural circumstances. So there's a lot of the conclusions we draw from these are also, they're useful, but they're a bit standalone, and you need this sort of wild populations also on the side where you get other information from, and then you can synthesize. Anyways, to study aging in wild populations, you need repeated measures on individuals, you need to know accurate ages, and you need to know accurate reproductive success or fitness data. And as I've explained before, we have that on Lundy, so that is a very good data set to study aging in wild populations. And the first thing I've done quite a while ago now is I've looked at reproductive senescence, at whether they do actually change over the course of their years, in how many offspring they have every year. And what you find is, and that is the number of annual growths on the y-axis and the age of the sparrow on the x-axis. And you find that in the first year, they're not so good. We do like 2. And then that improves, and it does a hump, and then reproduce best between 3 and 4 years of age. And the same is actually true if you look at the number of recruits. It's just that they reach their peak a bit earlier, and they peak for a bit longer. And I think the difference between these two graphs can be explained by that they improve in parenting, in actually doing that, feeding their young and doing all these things. And I think that is what you see, that they, in the second year, they're already good enough to kind of raise their young to then survive for the next year. So sparrows do show senescence, but and their age. So the important bit here is, of course, not the improvement in the beginning, but this decline afterwards, which is really a biological decline. And the older they get, they don't do that very well. By the way, which makes it really weird why cheating females prefer older males, because they actually don't provide any benefits in that case either. But hey, but of course now it's interesting to look at the extrinsic versus intrinsic mortality, and also to look at whether Lundy is a bit of a special case or whether there is something that all sparrows do. And what we've done is we've compared mortality. If you remember, we have this extrinsic mortality and intrinsic mortality, and we can look at these things also in this data. There's statistical ways of disentangling that. And what we looked at is, Merit did that, postdoc. He looked at that in my captive birds and in the wild birds and compared that. And the reasoning was that you would expect that the captive birds have a lower catastrophic mortality, but you would hope that the rest is the same. And that would mean that there's some sort of real, intrinsic biological aging going on in sparrows. And this is what he's done. So the dashed line, the upper line is the wild population, this is all mortality, and the lower line is the captive population. And I need to explain this bit to you. And that is the mortality, or the mortality rate on the y-axis. On the x-axis is again age in years. You can see the captive birds, they live a bit longer, so we have better data there from 1 to 9 years. But what you see here is that the wild population is higher. That means they have a higher mortality. And the level at what height they are is the catastrophic mortality. So that's what you would expect. In the wild, there's more stuff happening, like sparrowhawk coming through and killing them. But the slope, the amount of increase here by age is the same for wild versus for captive birds, and that's the intrinsic mortality. And that means that each year that they age, the risk of dying increases by the same amount for wild as in captive birds. And that means that biologically something happens inside the bodies that makes them more likely to die when they age. And that is the same process in wild as it is in captive birds. I thought that was quite fascinating. So this is consistent between wild and captive birds, which is quite nice. And that means that there is indeed senescence in sparrows. I did look at whether how long they live is heritable, and we found no, which was a bit of a surprise, because we thought, well, typically, individuals that live long have offspring that also live long. But then we also found something that was weird. We found that reproductive fitness was heritable. We found, at least, even though it was a small amount, but we did find that birds that produce a lot of offspring annually would produce offspring and also produce a lot of offspring annually. And that is really weird, because in evolution, we think this shouldn't be, because again, that could create this sort of Darwinian demon that I spoke to you earlier. And so I had a bit of a better look at that, and I found that there were some maternal effects, and maternal effects are influences of the mother that are not genetic. I know that's really complex and really weird, and we don't really know how that works, but we can find that pattern statistically. And I just thought, I couldn't make head nor tail out of this. And where that led me is it led me to thinking about maybe it's stress. Sometimes it can go through the egg for hormones in the egg. So if the mother has a lot of stress, she might pass that on to her offspring. And we know, and this is a horrible picture, I know this woman died, it's in the Ukraine, where this woman was pregnant, and then where she lived was bombed. And I just thought, this is such a traumatic thing to happen, not only to the woman, but also to the child. And we know actually that our ancestors' traumatic past has an effect on us. And I don't know why this moves on by itself, but there's, so this is my great grandmother who's lived through 2 world wars, has been displaced. And this is a daughter who has been actually raped by Russians during the displacement, then married, had a daughter, and lost her husband when her daughter was 6 weeks old, and then raised her as a single displaced woman. And then my mother, me and my daughter. And there is knowledge about when about how these sort of traumatic experiences can really be inherited to offspring through non-genetic mechanisms. And the evidence we have comes from when the Germans invaded the Netherlands, and the Dutch had to go through one horrible, horrible winter where they didn't have any food, where they couldn't get any food in, and it's called a hunger winter of the Dutch. And in this winter, when you take women who were pregnant during this starvation phase, and when you look at their offspring, at their children and at their grandchildren, you will find patterns that are reliable and consistent. And you will find that after a traumatic event, and that can be anything, it can be food limitation, chemical exposure, radiation exposure, stress, and also age, because a lot of the symptoms of stress mirror the symptoms of age. And what you find then is that in the offspring, you have a higher risk of diabetes, cardiovascular disease, higher mortality, there's differences in brain structure and size, there are behavioral changes, there are physiological changes. And in the great offspring, you still find things like this, apologies, and you even find a higher chance of having depression. So there is our traumatic, our ancestors' traumatic past can affect us, but we don't know about the mechanism, but there's a lot of evidence for this. There's also evidence, not only in humans, but there's some evidence in mice, in lab rats, in insects, in Drosophila, in yeast, even in some plants, in duckweed. We have this evidence for a transgenerational effect, either of age or stress or traumatic events. And we really don't know how that works. And I thought maybe we have that in the sparrows. So of course I had a look because we have that data. So what we find is sparrows. So there's a lifetime number of recruits, and it's called predicted because it comes from a model that looks a bit cleaner, and the age of the mother and the age of the father. What we find is that sparrows born to older parents have lower lifetime fitness. So throughout their lives, they produce less offspring when their parents was older compared to their siblings born to when their parents were younger. Usually when I tell this, people kind of start doing all sorts of mental gymnastics in that. I've also found that for grandparent age, the maternal grandparent line in particular, And that was really, and when I started doing this, people would say like, oh yeah, you know, they're just crappy parents. When they get older, they just start slacking. Well, we've done this cross-fostering experiment, so I could look at genetic age and cross-foster age, and what we found was it is definitely the genetic age that produces that effect. So even sparrows born to old parents but raised by young parents show that effect, which is again a thing you can't do with humans for whatever reason, you know. So in humans it's difficult because, you know, humans are traumatized. Traumatized humans raise their children differently from non-traumatized humans. So how can you say? But in this case, you can really, there's just the egg, nothing else. There's a transmission going on from parent to offspring. I don't know. So it's really, so how does that even work? It's like this, I don't know. There's something that needs to go into the sperm and egg cell that then goes into the offspring. But because the genes stay the same, they're immutable. How you know, it's a bit fantastic when you think about that. So there's something that happens here, and we don't know what. Potential pathways. There could be damage to the DNA, like the egg or the sperm. There could be something about telomeres, and I'm going to explain to you what telomeres are. There could be something about methylation. I'm not talking about this because the results are not really solid yet, and I don't want to go into a whole lecture about that. But it could also have to do something with the fluid of the ejaculate, because that changes in males, right? That is produced every time a male mates with a female, again and again and again. So maybe there is something. So it's really bizarre. So let's talk about telomeres. Telomeres are really, really interesting, I think. Telomeres are the end caps of the chromosomes. So what is nicely lit up green here. So these chromosomes are in our cells that where our DNA is. Let's have a closer look at that. So you have the cell, you have the nucleus, and in there are the chromosomes, and at the end of the chromosomes are the telomeres. So telomeres are really just genetic information that are repeats of always the same bases, and that is what we call telomeres. And they have a function, and there's a reason they are at the end of it. So this is a telomere of an adult cell. What happens when the cell replicates is, you have to think about it as like a zipper, right? The zipper runs across the genetic code. And at some point it has to stop and go back, right? But how does it know that it does that without kind of falling off? It hits the telomere. And then it goes the other way around. But the thing is, whenever it hits the end, it cuts off a bit. And it goes to the other side and it replicates again, cuts it off. So every time a cell multiplies, the end caps, the telomere end caps shrink. And now the idea is, when you age, when you hit that point where the bodily functions kind of decrease, is where the telomeres are depleted, because at some point they're gone, right? And we can show that. We can actually measure the length of telomeres, and in many organisms that correlates with lifespan, which is a bit creepy, and which also led to a lot of weird companies going through, we measure your telomeres and tell you when you'll die. It doesn't work like that. It would be nice, but it doesn't, because there's also a thing called telomerase that can make them longer again. So that happens, but there's always a but. The problem is telomerase sometimes goes out of control, and when it goes out of control, that is when cancer happens. You see, so there's this sort of balance, and that seems to kind of drive our aging process and our mortality. So telomeres are at the heart of that. They shorten with cell replication, they are correlated with future survival and longevity, and they can be elongated through telomerase, which, when it gets out of hand, causes cancer. So it's this whole weird thing that is correlated, but we're not so sure how causally related it is to aging, but it is excellent biomarker of biological age. So you can measure your telomeres. If they're short, we can say you've had a lot of stress in your life. That is what we can say. So we can use them in animals, obviously, to kind of learn more about this and figure out what they really tell us. This is Janet. The first thing she did for a PhD is that she did a meta-analysis where she pulled together all studies on telomere heritability. Because of course, when we look at these transgenerational effects, we want to see, well, what happens? And if older parents have shorter telomeres, maybe they can inherit that in a way. And she found there's a moderate amount of heritability. So yes, there is heritability, but it differs by species. So again, some of them are significant, some of them are not. So there's all a bit like, yeah, maybe, but not a kind of solid answer, right? And we think the reason for why this is so variable may be because the ages of the individuals may not have been taken into account. So when we look at telomeres in the Lundy sparrows, I said we had a lot of samples of adults, and when we look at the telomere length on the y-axis and the age, it says centered age because it's a within individual analysis. Remember, I think we talked about that. What you find is, yes, they shorten to life. There's a lot of variation, and they do predict survival in house sparrows, which is quite cool. And there is some heritability also to this. So maybe they can act as some sort of vehicle. Next thing Janet did, is she's really clever. She does these statistical models where I go like, I have no clue what you've just done. She's really good at that. And what she did is she looked at whether the heritability, how well or not well, the length of the telomere is inherited, changes with age. And it is called genetic by age effect. And this is the most complex thing I hope I'm going to explain to you tonight. So if you get this, you're good. So what she finds is that in middle age, there's not a lot of heritability, so not much happens. But the older they get, the more alike are the telomeres of the offspring to the parents. So that might be, even if it's not a causal process, but it might be an indicator, something that is correlated to both, that could drive these transgenerational effects. This is really cool. This has never been shown before anywhere. So I'm just so if you're looking at that, and if it's published online, because you asked me, is that okay? Just please don't, don't kind of promote that because we're about to publish it. But we also found there are some transgenerational effects with the telomeres, because of course that's the next thing we looked at, and Aaron looked at that, and we looked at within individual effects, and we found that young males produce offspring with longer telomeres, and old males produce offspring with shorter telomeres. And we're like, okay, that makes sense, right? When they're older, they produce less good offspring, because if they have shorter telomeres, they have less good prospects. And we thought that was nice. And the next step that we've done, is we've done an experiment, because you can't just, we had that before, right? You always need to do an experiment to show these sort of really important things. And what we've done is, again, it's really difficult to tell a sparrow, okay, look, you can only reproduce with an old male. It's like, so you have to kind of come up with a way. And what we've done is we, again, made aviaries by age this time, where there were only old males and young females. And then we had one with old males and old females, and one with young females and old, but you get the gist, right? So they didn't have a choice. They had a choice of partner, but they couldn't choose how old they were. And then we measured the telomeres, and Sophie did that. But that is in the aviaries. And what she finds was really weird. On y-axis, you've got the age of the father, whether it's young or old. On the x-axis is the telomere ratio, and the red and the blue shows whether it's a daughter or a son. And if you have trouble reading that, that's okay. Every time I look at that, I go like, oh, it takes me a while. But in short, what she found is that older fathers make daughters with shorter younger fathers make daughters with shorter telomeres, and older fathers make daughters with longer. So it's practically the opposite of what we found in the wild. It's big, okay. I need to finish some of it. It's not much. So that was really weird. And I don't know what that means. And I think there's some sort of optimization going on, where when you do an experiment, you find what's actually happening. While when you look in the wild, it might look different. I don't know. But, well, we don't know much about this yet. So it's 10 to. Do you want me to finish now, or do you want me to continue? Yeah. Because this is a good point. I don't want to go through this. So there is one thing when we think about how is this even work. I think this is something that I want to tell you before we finish, because there's a sex difference in timeline. Because there was this males and daughters and males and sons, and we always find these sort of differences in the sexes. And I think this is important to know. When you look at how sperm is produced, this is the whole thing. You don't need to know about all the differences, but the important bit is where the sperm, where the genes go into the sperm. And that happens all through males' lives. But in women, in females, that is different. What happens from puberty on, the whole process goes on and on and on. But in women, all this first part, where the genes are allocated into X cells, happens while the female is still in utero, in a mother's body. So I think what we really need to do is we need to look at differences in time and differences in sexes with these transgenerational effects to solve that. Because the timing of when things can change in the germline is different. I can talk a bit about mice, but I'll leave this. So in summary, telomere length might be important, and it varies, the heritability of that varies across age. Early life stress reduces longevity and fitness. I didn't show you that there's no effect with telomeres. And we still don't know the mechanism. We know that we can use telomeres good as biomarkers. We know something happens with small RNA, which I haven't shown you, but we still don't know how these transgenerational effects occur. So, and as a summary, this is from the first part. Social network is important for making decisions. It might explain why females take part in cheating. Wild birds age. Transgenerational effects occur, but we don't really know how and why, but we think telomeres are involved. And obviously I didn't do that work all by myself. There's lots and lots of students and field assistants and mentors involved. In particular, I want to mention Terry Burke up here, who has started the Lundy House Sparrow population together with Ian Owens and handed that over to me. And I want to thank you for your attention. Yeah.
Thank you, Julia. There's a lot to ponder on there. I would just quite like to ask a question though. Is the gene, there must be a lot of incest going on, mustn't there? If you think about it, surely they must be mating young, does that not affect the gene pool?
Yes, that affects the gene pool, but it's actually the population of Lundy is large enough that it doesn't. And because we have these regular immigrants, that kind of also keeps it fresh.
Right.
Yeah. So that is we can look at, but there is definitely more inbreeding than in the normal population. There will be, and this is why I use the capital birds to make sure there's generality of my claims.
And another thing that occurs to me actually generally as I'm going about my daily life is why you don't see dead birds lying around. But because you know your birds on the island, do you find them just lying dead when they died naturally? Because we don't see birds lying around. You don't see blackbirds, robins, crows just lying on the ground, do you? When they die, where do they go? So is that something you can see on the island?
Actually, yes. So you don't find them because they're eaten usually. As a cat that comes around, or like a raptor. That goes really fast, or also insects. That sort of process happens really fast. We find their birds in nest boxes sometimes when they go there and die there, because there's no mammal, like predatory mammal on Lundy. You've got sheep and stuff, but you don't have rats or cats or anything.
No.
So you find them more often. But if birds of prey reach them, they also get them.
Okay. Does anybody else have one very quick question before we wrap up the meeting? Anybody? Yes.
You mentioned bird flu. Are the sparrows affected at all by breeding influenza?
We've just been asked, are the sparrows affected at all by bird flu?
Yeah, I honestly don't know. I think yes. As I told you, the population has crashed now, but we have not been able to test for that yet. I would like to do that. Yeah. I know passerines are less affected than like seabirds, but
Anyway, Julia, I'm sorry that we've had to cut you short, a little bit short. I'm awfully sorry, but could we just thank Julia again for coming all the way from Bracknell to. And not to forget, everybody, that the next meeting is the Conservation Value of Ringing on the 18th of January, and on the 1st of February, the oh God, I can't read that again. Monitoring rare breeding species. Okay, thank you, everybody. Goodbye to everybody at home, and we'll see you next time.
thank you for having me.